Continuing our reading in the Cambridge World Prehistory, Volume 1, Africa, South and Southeast Asia, and the Pacific. It's edited by Colin Renfrew and Paul Bond, published by Cambridge University Press. We're continuing our introduction section, talking about DNA before we move on to languages in the introduction section. Concerning ancient DNA, archaeogeneticists are typically interested in tracing the prehistoric ancestry of living individuals. They seek, for example, to find out about the ancestors' arrival times in various parts of the world, or to find out when they developed specific features, such as depigmented skin, milk digestion in adulthood, or malaria resistance. To achieve this, it is necessary to analyze the DNA of living individuals rather than of prehistoric fossils, as there is no guarantee that any particular prehistoric fossil contributed genes to the present population. However, analyzing the DNA of fossil remains, along with ancillary organisms such as human parasites and domesticated plant and animal species, it is important to reconstruct the context in which our ancestors lived. Specifically, a fossil hominin DNA profile can tell us when that fossil's lineage split from our lineage and allow speculation as to how different genetically that hominin would have been from our human ancestors. Even data sets derived from modern DNA generally contain numerous errors, and it is therefore no surprise that errors are also a significant feature of ancient DNA datasets. Similarly, first attempts to obtain whole genome data from ancient Neanderthal DNA, for example, have had to be retracted due to contamination problems identified by J.D. Wall and S.K. Kemp. Ideally, ancient DNA data should be validated, and this can be achieved in a number of ways. For example, archaeogeneticists can type the DNA of unknown descendants of the ancient DNA sample. Another approach is the typing of accompanying animal bones to prove that the environmental conditions permitted ancient DNA survival. Another is typing of several prehistoric human individuals from the same context, in the expectation that they would systematically differ in their DNA from any modern DNA. Initial ancient DNA studies in the 1980s and 1990s were met with skepticism, coming to a head when successful retrieval of 80-million-year-old dinosaur DNA had been claimed and then shown to be modern DNA contamination. The first mtDNA sequence obtained from a Neanderthalensis was the first convincing and significant breakthrough in ancient DNA analysis, and incidentally did not require any of these controls, as the resulting sequence was on the one hand clearly non-human, and then on the other hand retained some ancient mtDNA variants predicted in some of the deepest African mtDNA branches of modern humans. The Neanderthal sequence by Krings and his colleagues, obtained from the Feldhofer-type fossil in the Neander Valley in western Germany, was soon validated by independent analyses of other Neanderthals, and showed that humans and Neanderthalensis had split approximately 500,000 years ago. This new timescale led to the popularization of the proposal that Homo neanderthalensis was related through an ancestral African Homo heidelbergensis to modern humans. Another important advance that came about through ancient DNA studies of Neanderthals is a better appreciation of their geographic range. Fossil bones in Central Asia that hitherto were too fragmentary for species assignment were shown by ancient DNA analysis to be Neanderthal, thus expanding the known range of Neanderthals eastwards by at least 2,000 kilometers. The picture might appear more complicated, however, with the ancient DNA analysis of other fossil fragment from the Caucasus Mountains, which reveals that the hominin in question is neither human nor Neanderthal, but split from the human and Neanderthal lineage some 1 million years ago, which might indicate an independent migration out of Africa. Subsequently, these researchers published whole genome sequencing of the Denisova tooth and phalanx, concluding that the Denisovans have evolutionary histories distinct from Western Eurasian Neanderthals and from modern humans, or anatomically modern humans, implying that Homo heidelbergensis gave rise to Neanderthals and Denisovans in the past 1 million years, both going extinct with the arrival of modern humans in the past 50,000 years. An interesting claim made by Reich and his colleagues is that 4 to 6% of modern Melanesian DNA derives from the Denisovans. The breakthrough with Neanderthalensis DNA set the stage for accepting ancient DNA results from consciously performed ancient DNA retrieval from anatomically modern humans. Although available modern human remains are generally younger than Neanderthal remains, it is much more challenging in such cases to prove that the retrieved DNA is generally from the ancient fossil rather than from a modern human contaminant. Nevertheless, by analyzing groups of prehistoric individuals, archaeogeneticists have obtained internally consistent results, starting with the analysis of the pre-Columbian Oneota Cemetery in North America by A.C. Stone and M. Stoneking. Another breakthrough for ancient DNA from anatomically modern humans came with the typing of mtDNA in Central Europe's first farmers by Joachim Berger and colleagues. The bones were from the linear Bonkeramic period and therefore between 7,000 and 7,500 years old, and the identified DNA types were convincingly ancient because it so happens that many of the first farmers had DNA types that today are exceedingly rare in Europe or indeed in the wider world. This study was followed by another study done by the same team to characterize the DNA of European hunter-gatherers. They analyzed the samples and they ranged from Germany over the time range from the late glacial Paleolithic period up to 15,000 years old at the one extreme to late Mesolithic hunter-gatherer samples from the Baltic states and Russia, some of which were only 4,500 years old. Again, the uniform DNA types found across this wide area and range of millennia validated the retrieval of genuine ancient DNA. Not long afterwards, the results were confirmed by independent teams in Sweden and Leipzig studying hunter-gatherers in Scandinavia and southern Russia, respectively. Details on these large, unexpected research results are given in Chapter 3.18 on Europe and the Mediterranean. For more recent times, ancient Central Asian DNA has been retrieved from permafrost burials dating from a few centuries to over 25,000 years old, where the collaborative team of Bertrand Lutz has been working. Here, validation is possible because the DNA preservation is so good that not only the mtDNA but also nuclear DNA was successfully retrieved, demonstrating that some neighboring skeletons have familial relationships and that the identified genetic sex corresponds to the morphological sex of the skeletons. The development of ancient DNA techniques offers an important avenue for shedding light on the descendant of ancient populations, since it should be possible to study different periods in the descent process. One avenue of research emerging from the genetic sections in the present book is the possible inference that early language change may have been influenced more significantly by the male component of an incoming population, as represented in the Y chromosome, than by the female component, seen in the mitochondrial DNA. For example, in India, evidence from the molecular genetic data for an incoming population within the last 10,000 years has been difficult to document. Possible candidates could be Y chromosomal subtypes of J2 and R1A, found in uppercase today at a frequency of 10 to 20%, which could correlate with components of an incoming earlier population from the Northwest, and therefore possibly with a Proto-Indo-European speaking population. It should be noted, however, that no corresponding mtDNA effect is seen. A similar case may be seen with the Austronesian languages, where the molecular genetic input by Austronesians into coastal New Guinea may be discerned both in the Y chromosome type O and in the mtDNA type B. But it is only the Y chromosomal component that appears to correlate strongly with a linguistic transition to Austronesian in New Guinea. Here, too, the incoming East Asian component results in a level of 16% in the current Austronesian speakers in New Guinea. It is interesting that such low levels of male immigration should correlate with such a significant linguistic outcome. And so that is languages. Not languages. That is the DNA part of the introduction. We'll briefly go over the languages section and how language can inform prehistory. From the Tower of Babel to the tales of the Aboriginal Dreamtime, we have long sought to account for our baffling diversity of tongues, or languages. Today, however, prehistorians can look to our linguistic diversity not just as an enigma to be resolved, but as, in fact, a rich source of data on our origins, or at least potentially so, for it remains a challenge to work out quite how we are to interpret the linguistic record of our past. The science of linguistics first began out of this desire to make sense of our linguistic diversity, and has taken great strides over the last 2 centuries. Even so, the nature of language and how it changes through time still frustrates us. We do not know when it was invented, although we can have a brief time frame of when it probably was invented. It does not even allow, for instance, a straightforward answer to what may seem the simplest question: exactly how many different languages does humanity speak? Typical estimate range is around 6,000 to 7,000 spoken today, usually with reference to the ethnologue classification. But in truth, such figures mask considerable inconsistency and imprecision. The very question is in fact somewhat simplistic, akin to asking an archaeologist to count how many human cultures there are, or a geneticist how many populations. The definition of a discrete language as opposed to a dialect or language family is a matter of principle, by no means always clear-cut. Across much of the globe, counting languages relies on arbitrary lines drawn through what, in reality, are dialect continua. Across much of China, India, the Native Americas, Arabic and Bantu-speaking countries, even Scandinavia. But by whatever measure, our linguistic diversity is vanishing at an alarming rate. Almost every week, 1 of 4 varieties of human language goes extinct, and each takes with it not just a unique repository of human culture, but also irreplaceable clues to our past. A first and striking example of how our languages can open up a window on prehistory lies in the broadest patterns in linguistic diversity worldwide. This illustration also helps explain why, for the general purposes of this chapter, exact language counts are in any case rather beside the point. For here, the aspect of diversity of most interest to us is the multiplicity, not of language units, but of independent or pristine language lineages, not demonstrably related to any other anywhere. Europe, for instance, hosts but 4 surviving lineages in this sense, recent immigration aside. Among them, both the most widespread of all language families, Indo-European and the single language, isolate. For example, a language not demonstrably related to any other worldwide. For example, South America and New Guinea, meanwhile, host many dozens of lineages apiece. Such striking differences cry out for explanations, which necessarily lie in the realm of prehistory. And not only are contrasts like the above immediately apparent, but so too are tantalizing hints of correspondences between that go beyond linguistics, with some of the defining oppositions in the real-world context that shaped the story of humanity: old world versus new, temperature zones versus tropics, agriculture versus hunting and gathering. Most proposed correlations remain but hypotheses, however, in heated cross-disciplinary dispute. Similarly, all proposals for taking the classification of our languages far back in time to putative macro families are mired in controversy. So, to try to sketch a clearer path through the world's language prehistory, we begin here with a return to first principles. We reconsider how it is that linguistics can inform us of the past in the first place, and what the basic methods and models are by which we may take better progress towards a new synthesis of the various disciplines with a stake in prehistory. And so in the next section, we will go over the introduction, primarily focusing on languages and how they can inform our understanding of prehistory.